Good afternoon, everyone, and welcome to our event on sex and gender disparities in medical research and practice. I hope you saw a bit what the MDC is about. Unfortunately, the sound was missing, but I think you still have an idea of what we do here. And I'm Lieke van der Haar. I'm a postdoctoral fellow here at the Berlin Institute of Health. Sorry, not Berlin Institute of Health, Berlin Institute of Medical Systems Biology here at the Max Delbrück Center. And as a scientist, I am interested in how human tumors develop. So I work together with clinicians here at the Charité, about 500 meters away from here. And the clinicians sent me human tumor samples. And I received these samples and I researched them here a few floors above, actually. And I nicely note down the sex of the different patients that I'm collecting. And I try to make an effort to kind of balance this out between men and women. But there my effort also stops. So I do not specifically look into this as an important variable in my research data, basically. And tonight we'll hear from 5 speakers who do see sex and gender as a significant variable in their research. And tonight we'll hope to convince you about the importance of gender medicine and, yeah, basically to show you about this. Also, we will have a panel discussion, and in this panel discussion, we'll touch upon subjects of, for example, are women the only ones that benefit from gender-sensitive medicine? And also, who are the major stakeholders in the field? Whose responsibility is it to bridge the knowledge gap regarding sex and gender in medicine? And next to that, we would also be very happy to hear from you here, the audience. So please feel free to ask questions after the presentations of the speakers. And also share your thoughts online. Please use the hashtags here so that we can follow your IDs and maybe do something with them later. And also I would like to remind you that this event is recorded. And with that, we start with a word of welcome from the scientific director of the Max Delbrück Center, Professor Dr. Maike Stadler.
Well, good afternoon, everyone, and welcome to the Berlin Science Week here at the Max Delbrück Center. I'm really pleased to see so many of you, and I know you come really also from diverse backgrounds. Some of you are medical researchers, others are social scientists, and then we also have journalists here to discuss today a really important and pressing issue in science, which is diversity in research. And we know that there are really important implications for improved research and medical care for everyone. So today's event will be mainly about sex and gender issues, but I want to lead off by saying that actually the issue of diversity in research extends even beyond gender and sex issues, of course, to much more, to different ethnicities, different physical and mental abilities, and then also our socioeconomic status, to just name a few. And all of this is now rising really to the forefront of research, and that is really important. So the next frontier, when we think about where we are in medicine and where maybe the next sort of leap in really improving medical care comes from, is really that we're now living right at that era where we're beginning to implement precision medicine. So we already have like a few examples. For example, you can now, when you are diagnosed with breast cancer, you can undergo a screening of the tumor, and then the therapy will be specifically tailored to the kind of tumor you have. Or we have also this amazing example of T-cell therapies that we are now using for hematological cancers. Some of these technologies are actually being developed here at the MDC. And that also represents an example for really sort of a personalized type of precision medicine. And then there is HIV treatment, where we can now really look at a specific individual's a profile of the viral strain that they're infected with, and then really tailor also the therapy to that. So where is really that next challenge, and where do sort of gender and sex and ethnicity and other dimensions of diversity factor now into our research and developing new therapies? And one really sort of next thing is pharmacogenomics. So what that is that, you know, maybe in like 5 or 10 years' time, you could walk into a doctor's office, get your genome profiled, and then with that profile, that would be able to predict what kind of drug would actually be the right drug for you. So we would then link really our individual genetics to our ability to respond to a certain treatment. So can we already implement that? There are actually many sort of caveats and gaps in our knowledge. And especially in the field of genetics, a lot of sort of linking our genomes to disease risk and now also to the linking it to therapeutic options has all been done in Caucasians. And at least, and also for most initially Caucasian males. But there is really a huge gap that we also need to now look at other ethnicities, because what we know is that what the kind of treatment that might work well in a Caucasian might actually not work in somebody of African descent or Asian descent, and we're seeing that all over. So we know there is a huge need, and I want to share with you a little bit of sort of my experience from my own field of research. So I work in diabetes research, and I can attest to that the lab research is actually mostly done in male animal models. So most of what we know is actually all been tested, all the mechanisms that we research to what lowers blood sugar and how it's controlled is all been studied sort of in male mice or male rats. But we actually know for some research that sex hormones like androgens and estrogens really affect how our beta cells work. And the beta cells are the cells in the body that produce insulin, and insulin is the hormone that ultimately allows us to metabolize sugar after a meal. So one most startling really example of also the importance of ethnicity that I, even as a diabetes researcher, didn't know that much about, but learned about at a visit to Winnipeg a year ago, is how important actually ethnicity is also for diabetes research. So it's well known that people of Inuit descent, so people who have lived in the Canadian North or in the Arctic in general, have a very, very high rate of diabetes. And one colleague of mine in Winnipeg, who leads the diabetes center there, is actually trying to understand in her research exactly why that is. And what she is finding is when she's taking beta cells from people of Inuit descent, that they respond entirely differently to like the normal sort of factors that stimulate insulin secretion, like glucose, fatty acids, or proteins. And when you step back, that actually makes perfect sense, because people of Inuit descent adapted to a diet that is just fat and protein. When you're out in the Arctic, there are no carbohydrates, there is no grain. And then with the colonization of Canada, all the people actually then had to sort of migrate south, and they were actually exposed now to carbohydrate-rich diets. And their bodies simply cannot metabolize carbohydrates. And about 80% of the Inuit population in Winnipeg actually has type 2 diabetes. So that actually taking that a step further, the normal drugs that, again, were all tested for diabetes in people of Caucasian descent also don't work well in that population. And there I'm seeing like this really important link that the research that my colleague is doing there now is beginning to actually really understand how does a beta cell of a person with Inuit descent actually work, and compare that to somebody who's Caucasian, and that will also allow us to close that gap for finding new drugs. So what is needed to really close that research gap right now to make sure that precision medicine arrives really for all of us, that not only people of a certain descent and ethnicity benefit from all this research that we are doing, and I want to comment on what the MDC is doing. So one thing that we clearly need more diversity of is also diversity in the workforce and study subjects. And I think the two things are actually linked, because as we have sort of more diverse teams in our workforce, there's also, we're going to raise awareness as to how important that is, because we all know that if we don't have people at the table and they're not represented, most likely we will also not really consider their needs in our research. So there are, thankfully, also large-scale efforts already underway. In the US, they started at the National Institutes of Health, this All of Us research program, where now they're beginning to stratify and really categorize people based on population-specific risk factors, on their different backgrounds, and then they're feeding that knowledge and really trying to recommend the right clinical trials to people. So we're beginning to actually customize our clinical trials landscape. So we at the MDC, we are also really interested in broadening our approach and making our research more diverse. And we have a collaboration, as one example, with Stellenbosch University, where we're now reaching out to that university, and they're really interested in also understanding how the genomes of people of African descent function. And we're hoping to really link up our research to also that knowledge base to really broaden also the approaches that we take and widening the kinds of insights that we're going to gain. Then we have a collaboration with the Nova Institute in Portugal. We're actually building a sister institute there in Lisbon. And they have a huge global health program and also a huge focus on Africa. And then we're hoping to bring some of these technologies that we're using here to research and really the mechanisms of disease and bringing that again to a wider population. So just to conclude, to improve human health for all and implement precision medicine, we need clearly more diversity in research. So we need to understand the unique environmental and genetic factors that impact risk for disease and our response to therapy. So events like today will kickstart this long overdue dialogue about how we can make research more diverse and inclusive. So the MDC has clearly committed to being a thought leader in this effort. So I'm really proud to also host this event here today. And I want to really thank all the speakers who have agreed to speak here today. And they're going to share with you their insight on these important questions. And I much look forward to hearing what they actually have to say in the next 2 hours. So welcome to the event. And the next speaker is, I actually have to... Sofia. So I will welcome Sofia Forslund to the stage.
I think that was a very good summary of why diversity in science is so important, and it's also great to hear about the efforts that are already being undertaken here at the MDC. And indeed, our next speaker is Professor Dr. Sofia Forslund Startseva. And she is a computational biologist and professor for applied microbiology at the Charité. She's also the head of a research group called Host Microbiome Factors in Cardiovascular Disease here at the Max Delbruck Center. And originally from Sweden, she realized quickly in her life that mathematics is in everything around her. And for the last decade, she has worked on omics analysis to investigate how the diverse array of bacteria in the gut affect our health and vice versa. And her focus is specifically on the host microbiome interactions in the context of cardiovascular and metabolic disorders. And her interest in gender medicine comes both from being a feminist, a trans woman, but also as someone with a general scientific interest in overarching master regulators of biological processes, such as those within human endocrinology. Sofia, the floor is yours.
Thank you so much. Thank you so much again to Maike for a wonderful introduction, which I think really starts this very well, because I would like to continue to sort of underscore how I feel that gender medicine is an exciting frontier of personalized or precision medicine, and one where we can go more granularly and more individually than has perhaps so far been done. I'm not going to go into a lot of details about anything we found, but I will describe some of the approaches we're taking to try to get closer to useful insights in these regards with some examples from what we've been doing. See if this works. Yes. Now, as you know, also from the introduction, but I'm sure also generally since you're here, most medicine so far has been researched and practiced assuming men as a norm that has been adapted to. So we've overlooked, both with regards to research and medicine, situations where either men or women are more at risk, respond worse to treatment, or show different symptoms. One example being of heart attack symptoms. There are a number of examples where this is relevant. Some are quite obvious. For example, there are cancers affecting reproductive systems, breast cancers, prostate cancers, cervical cancers, and so on, or otherwise affecting those tissues, like in case of endometriosis or preeclampsia. But there are also other less evident situations, like, for example, as Maike was mentioning, factors in diabetes, in heart diseases, autoimmune diseases, where we also see differences between groups of men and groups of women, for example, in these regards. Just to give you an example, just more clearly what direction I come from, is basically we try to understand cardiovascular, metabolic, and autoimmune diseases, not because we are medical specialists, but because we are interested in trying to see, can we find medically relevant things from doing large-scale, multidimensional measurements on human bodies or animals over time and in the course of treatments, then put those into computers and get something out that we can model and that we can draw conclusions from, mainly from the perspective of answering the question you saw in the very beginning of the movie. So why do some people get sick and some people get healthy? Why do some people respond better to some medications than others? Just seeing, can we analyze data coming from interesting populations or interesting treatment cases or interesting experiments to get useful insights? Getting into the area of gender medicine, it does become relevant to ask, what do we mean when we say that a person is a man or a woman? Now, my native Swedish, just as German, doesn't separate sex and gender, and I think there's a utility in that, but in English this is frequently the case. If we do that, my preference would be to let gender be a term that describes a life and sex be a term that describes the properties of a body. So a life is gendered and a body is sexed. More specifically, for example, lives would be gendered by men and women. For example, having different experiences, living in different situations, different power situations, and therefore being exposed to different environmental risks in ways that can also differ a lot between different cultures and across history. This includes things like stress levels, dietary patterns, drug use, and so on, with certainly major health impacts. But what I'm mainly focusing on here isn't that gendered lifestyle risk factor, but the aspects of the sex properties of the bodies and how they impact our medical situation. And I would ultimately, as someone coming from an evolutionary science background, say that sexed aspects of bodies are aspects of the biology of bodies that reflect evolutionary adaptations to how we participate in processes of sexual reproduction in one way or the other. In many cases, very indirect such adaptations. And there are a number of ways in which bodies are sexed that I would point out here that also in principle can sometimes vary independently, starting from what most people are aware of, like the chromosomal complement of each cell, the karyotype, which in humans and some other mammals is the XY chromosome set up for sex determination. But also what genes are on those chromosomes in a given person, which of those genes are set in semi-permanent or permanent on or off states through epigenetic modifications, which genetic programs are active in different tissues at different life stages, for example, driven by hormonal factors which can change across a lifetime. Certainly things like the gonads, the organs that mainly produce sex hormones, their presence and activity across a lifetime. Ultimately, all these things contributing to build and maintain a body over a lifespan, which has an anatomy and which has a physiology, which is how that body functions and what functions that body carries out, and then interacts with its environment, living that gendered life. There is relevance here, I think, if we talk about biological sex for a body or a cell or a tissue. we are actually summarizing this large-scale factor of many different components in a way that's useful for our context. I really like the phrasing that we used in this paper in Science a few years back, saying that sex is a context-dependent summary of a multidimensional variable space, where I'd focus on the context-dependent in that we are speaking of these things for a purpose. In this case, we do so because we want to help people stay healthy and live long lives. So for a medical purpose of preventing and trying to treat diseases, what we refer to when we speak of biological sex are a certain set of processes relevant for that purpose. In most people who are cisgender or cissexual, these are all correlated features, like they stay more or less the same throughout. Can I even do that, or do I need to? Yeah. I don't think I have a pointer, but that's okay. Mainly, now my point is that in most human beings, these factors all line up. In animals, we can't necessarily speak of a gender because we can't talk to them, but other factors usually line up. This is useful because it means in most cases, in a healthcare situation, you come across a person, you can easily come up with sort of what is most likely to work for them. But it does make studying the underlying mechanisms more difficult. Because if we see, for example, in one project we've got going, that men more often have heart failure with reduced ejection fraction, is that because they eat more meat or stress more or smoke more? Is it because of testosterone effects, or is it because there's something on the X chromosome that's protective if you have 2 copies of it, or something else? We cannot just compare groups of men and women and see this. There are ways in which we can do this experimentally. So one easy approach if you want, is to do things to animals, modify them genetically, modify them surgically, modify them pharmaceutically, and then we can change one or more of these factors separately out of alignment with the rest of them and create models for more complicated situations. One example being removing the gonads and replacing hormones with exogenous administration. Another being we can move away the testis determining factor from Y chromosome, place it somewhere else so we can still have the sort of XX complement, etc., which has led us to a number of interesting insights. For example, a lot, but not all of the differences seen between populations of cis men and cis women is traceable to sex hormone action. A bunch of things is not, different in different tissues, different in different life stages. However, there's only so far we can go with animals, because even though even the smallest mouse is terribly complex, there's still hundreds of generations of lifespan of mice for one given human lifespan. We develop far longer. We have thousands of times more cells, and we live far more variable lives. In principle, much of what we can say in a mouse can translate to the human, but much cannot, and we can't quite be sure of what, including whether or not we are, for example, more plastic in some cases throughout this far longer lifespan or not. So one thing one can do is sort of the natural experiment. There are humans that are born or developing due to factors during fetal development or later into intersex states of different kinds, where there's also some factors of their body that is not in alignment with the other factors, one way or the other, however these individuals define themselves, which can vary a lot also. But while there's been attempts at observing this is rare enough and diverse enough that finding volunteers from these populations for studying just one factor at a time is still not really realistic for what we would like to understand better in the mechanisms underlying sex implications in medicine, for example. Plus, we still end up having to compare one group of people to the other. We can't take just one component at a time and change it. So what we are trying to do in order to get around this relies on another population, namely transgender volunteers. So many transgender people like myself essentially experience distress with the sex we were assigned at birth, and so we try to do whatever we can to change the aspects of our sex through medical interventions. We have a few different options to do so, but the most accessible one is to change the sex hormone action in adulthood, which will change some of these factors, but not others. Other things you can do include surgical interventions. That's not what we're primarily looking at now, however. The process of changing sex hormone impact in adulthood in a transgender person is somewhat streamlined. It is a somewhat recurring thing happening, so especially in Berlin, which has a long history of being open to LGBT persons since Hirschfeld and forward, we're hoping to find enough people who might be able to help us by observing their journeys to get closer to an understanding like that. So we applied for funding to the German Cardiovascular Research Center to run a study doing exactly that, following people undergoing hormone transition therapy over a few years, following perhaps a 2-year span or longer if we get more funding for these individuals in either direction, adding, removing testosterone and adding estrogen, not actually removing estrogen, but it sort of fades on its own with ovarian action go down, adding testosterone. Following then what we can measure in terms of microbiome, in terms of gene expression, in terms of phenotype, cardiovascular risk factors of different kind, direct or indirect, to try to see, do we see the same patterns repeated and emerging as we see when comparing populations of cis men or cis women? I'm really excited to see where this is going to lead us, because it does mean we can get a little bit closer perhaps to the actual mechanisms underlying and how they can personalize. We hope to do several things this way, and one is, I know I'm a little bit over, I'm going to try to be as quick as I can, sorry about that. One is to get a better understanding of the way that the health implications of sex play out in each person. So again, the precision medicine angle. If a person does not match to sort of immediate first baseline expectations, what happens to them then? And this is more important perhaps than one can immediately appreciate, because these factors do change over a lifetime, and there are people born who are different in one way or another with regards to the different components of sex, and their healthcare needs are far less well elucidated and considered. And of course, one other factor is to better understand how we can meet the healthcare needs of trans people, including both are there ways we can mitigate any risk that might emerge in the course of treatment, and are there ways we can make that treatment more effective, understand why it works better in some people than others. So, for example, we want to try to also characterize this in other groups, similarly using a same streamlined phenotyping data collection measurements we would love to do, and we are in some cases looking at the menopause transition. We're looking also ideally in various types of pregnancy cohorts, including at-risk pregnancy cohorts. And I'd love to do studies into things like polycystic ovarian syndrome or endometriosis, or in individuals who have particularly severe menstrual cycles with side effects thereof, and try to see, can we see common patterns that we can understand better by understanding these factors of variation separately. So that is a few examples how I think we can really get closer to an understanding that benefits everyone by understanding the mechanisms better and by exploring the variation that we ourselves induce as we are alive. And I also want, again, to underscore, I think it's really important to note that this is, again, an area where the sex of a body is not always just one single specific essential thing, but it is a summary of many different properties that usually are aligned. I really like this paper also from a few years back that discusses this in the context of various kind of animal model research or cell model research. I think lays out a very useful pragmatic perspective to how to build better models for different diseases while not flattening that diversity too much. And I think for me personally, this is also important because we are at the point in history where on one hand, people like me are more visible and more accepted, less stigmatized than ever before. But there's also a lot of discourse going around that our biological sex remains what it was assigned at birth, and therefore we should be excluded from some areas of society, etc. And my response to that would be that biological sex is not just one thing. It is a complex set of things, and it depends why we are talking about it. It also depends on why we do what we do with it and how that changes in the course of a lifetime to determine what our needs are. I would want us to consider sex in the purely biological sense as being essentially that complex set of properties that we define for a purpose, and that purpose should be to make our lives as long and fruitful and thriving as possible, in particular to be able to meet our healthcare needs as well as possible. And those healthcare needs will be a certain set of needs for one person and another. They will change sometimes during a lifetime, during reproductive life core history. They will be different for people who are exceptional in some regards from birth or otherwise. I mean, a body like mine, for example, cannot get pregnant, has XY chromosomes, but I still need gynecology care, I still need breast cancer screening, etc. So usually, in most cases, I benefit from being considered as any other woman in case of healthcare system interactions, as long as it's borne in mind that no, I don't actually need lead shielding because I will not get pregnant when they do an X-ray. So generally speaking, a lot of things are not known about how we consider minority populations, and perhaps that can never be fully set down in stone, but needs to always be handled by exception. But I believe and hope that this kind of research can, in addition to better understanding the underlying mechanisms of both the mainstream and the fringes of sexual differentiation of humans, can shed some light on how we can better handle a wide range of populations with regards to their healthcare needs. So I will stop there. We can discuss a bit if there's some time, I think a little bit time at least, and then we can discuss more in the panel, and of course we can discuss in other venues and forums. And if any of you are considering starting hormone replacement therapy for transition in the next 6 months or so, we are definitely recruiting participants for this study, so in that case reach out before you do that so we can follow you as well. Thank you so much.
Are there questions from the audience for Professor Forslund? If not, you can also... I see one question there. Yes. Wait for the microphone, maybe. I
think I can speak
okay, fine.
Can everybody hear me? Yes. Okay, cool. So thank you for this very interesting talk that was even interesting for people who don't have like a PhD in medicine, like me. My question would be like, you have been working on this subject for many years. What was the largest surprise for you in this research process, looking on the topic of sex from a contextualizing perspective?
Actually, I didn't really look into this much before coming to Berlin, which I became more interested as I started transitioning myself. What has surprised me most, I think, is that there are a number of non-hormonal effects, including epigenetic changes happening very early in fetal development on both the autosomes and the sex chromosomes. And I think we need more careful evaluation of when and where these have effect and to what extent that they are also changing over time over longer time frames. So that it is more complicated also on the biological level and involve more mechanisms than any reductive stance would lead me to.
I also can talk louder. I'd like to ask a bit. You showed us a proposal that was awarded for that research, and I found it can often be difficult to be awarded or achieve funding in this area, particularly with it being understudied to begin with, and I think still a bit undervalued in some ways. So I wanted to see if you had any advice with approaching this to a scientific audience for funding.
I think we've had great advances in last decade perhaps, in that funding bodies are recognizing the sex and gender disparity. So we set aside funding for that and they designate that as a criterion that should be taken into account. Most people who apply for research funding, they do a sort of, what do you call it, cosmetic job of that. They say, yes, we will consider this, and then they don't do anything. So actually thoroughly, fully basing it on that with a bold methodology might be a way to go. Another factor, of course, is to consider how large populations are potentially affected, because ultimately healthcare funders will want to be interested in how can we reduce healthcare costs over the next decades or centuries for aging populations. So I think there is a willingness to get somewhere. The larger group of people is affected, the better, and the bolder the approach from a methodological sense, the better. Unfortunately, also generally in research, the more you've already done, the easier it is to get funding for more, which is unfair and shouldn't be the case. But you can in a way cheat that by collaborating with people who have a track record.
In case there's more questions for Professor Forslund, please find her after the event. She will be around, and you can ask your questions then. We'll go on to the next speaker. That's Dr. Claudia Crocini. And she leads a research group funded by the German Center for Cardiovascular Research at the Max Rubner Center at the Charité. And with her team, she is studying how male and female heart cells are different. And she advocates for incorporating sex as a critical variable, also in basically early research, a very fundamental research. And with this approach, Claudia aims to uncover unique characteristics of male and female heart cells, ultimately accelerating later stages of research and paving the way for a more tailored and effective treatment plan.
Thank you. Well, thanks a lot for the introduction and for including me in this fantastic event. I'm really glad to give like a basic scientist perspective, and I went a little bit kind of maybe basic science or also kind of easy, but I hope, I mean, if you have more questions, we can talk more about this. But before starting, I would like to kind of point out where are sex and gender in basic science. So we have it, of course, in our subject of observation, and I will mainly talk about that. But this is something also already came out. We also have it in the observer. And when I mean, when I say the observer, I mean not only research and scientists, but also, and probably more importantly, reviewers, editors, publishers, as well as funding agencies and politics, because this is where the money comes from, and so the real change will happen at this level. But as I said, I mean, this I think it's an important point of discussion, but I kind of would like to focus on the subjects of observation. And so as basic scientists, we can study a whole organism or a mouse model, let's say, or animal models, or humans. We can go deep into maybe one organ specifically, or even single cells, or even further down to single molecules, let's say a gene or proteins. And I would like to give you a couple of examples where I think studying sex has really been, of course, very important, or at least it was very surprising for me to see these differences. And also a case, I would start with a case where sex and gender differences are seen in humans in a way that it's kind of becoming the opposite of what we initially thought. And this is the case of ultramarathons. Ultramarathons are these, crazy races where people run way beyond the marathon distance, which is 42 kilometers. And women are continuously winning lately, at least in these kind of competitions. And this is the case of Courtney Dauwalter. I just wanted to show her because she, until 2017, was a middle school teacher and only went professional after that. And she's really smashing every record right now. And she's not the only one. There are quite a few others, women, also quite a few other men. But it's interesting because also women really don't participate to this kind of competitions. So the participation is really low, and still they are competing at the same level or even better. And so this is, the difference between male and female in sport and performance has been known for quite a while, but it's always been the case that we thought, okay, men are definitely faster or better at running, and so this is kind of a conception, but things are kind of changing, and already in It doesn't work. So in 1997, there was this study where they kind of compare the speed of men and women running as a function of the race distance. And as you can see, yes, it's true, a shorter distance, male are definitely winning, or they're definitely faster than females. But this difference really kind of goes away as we increase the race distance, and even goes to the break-even point is 66 kilometers, and you can see here where marathon is 42 kilometers, and really here the difference is not that much. So the study stops at 90 kilometers here. This ultramarathon can be in over 250 kilometers, so we really don't know what happens beyond that, but maybe female have an advantage here. Another kind of point that I thought it was really fascinating, it was the idea of the number of cells that populate an organ, and this can be somewhat, it turns out it can change between male and female. So we know, I mean, the heart can be, it's populated by different cell types. So we have the muscle cells, but we also have cells that, I don't know, kind of make our coronaries or arteries, or like immune cells and so on. Turns out that male and female hearts from humans don't have the same proportion of these cells. And this is, I thought it was really fascinating. So you see the red proportion, the red population, these are the muscle cells, while instead the yellow, for instance, are the endothelial cells, so the cells of Vaso. And those seems to be quite different in terms of proportion between male and females. And this is important because if you have a drug that specifically, targets the red population or the yellow population, we might see something really different. Between men and women, of course. So I thought that was very fascinating. And the other thing that I also found fascinating is that gene expression really changes a lot, even the healthy heart. So here you see genes kind of piled on top of each other and divided by chromosomes. So from chromosome number 1 to 22 are the autosomes, and then we have the sex chromosomes. And there is a bunch of genes that are not different between male and female, and these are the gray ones, and the black ones instead are the number of genes that are different. This is about 1,000, at least in this study. And so first of all, I thought it was also already kind of interesting that these differences happen also in a lot of chromosomes that are not the sex chromosomes. But of course, I mean, the obvious reaction would be, or at least it was for me, that, okay, those must be genes that are controlled by hormones, by sex hormones. And well, it's actually only 30% of them are under direct control of sex hormones. So there is a lot more going on, as we already heard from Sofia. And this is a huge opportunity for new discoveries, I think, which is really exciting. Let's see, what is the status of current sex and gender aware research? Well, I mean, this is a little bit of an older data set, but the situation has not changed that much, and it's a little bit frustrating, especially, so you see the females, these are the studies divided based on disciplines, and in blue is the female, and in green is the male. So these are the studies that only studied the female in blue and males in green. Some study both. This is the orange part, which is okay, it's not too bad. But then there is, I think this is the most frustrating part, about 20%, even 30% in cancer, that it's not even specified. So you read the whole article and you go through everything, and then we don't even know if their study was done in female and male. I think this is really a loss of information that could be maybe not used in this publication, but could be used later on from other scientists, for instance. So before ending, I would like to kind of maybe finish on a more positive note. So how can we accelerate maybe, or at least this is my perspective, how can we accelerate sex and gender aware research at the basic science level? And I like this approach that used some computational modeling, and also because I collaborate with one of those computational modelists from the University of California, Davis, and in her group they create computer models of cardiac cells that have just, the parameters are just a little bit tweaked based if they are female or male, so they can effectively create a male model of cardiac cell or a female model of cardiac cell. And then they found out that they can predict much better the response to drugs for both sexes, actually. So even having, a tailored male model will improve prediction for males. And of course, a huge improvement in prediction for females, since they have been neglected for so long. And also the other approach that they're using, I also think is quite interesting, is that they can translate from a male to the female and vice versa. So you measure something in a male cell, and then you put this information to a computer, and it will tell you, okay, then a female will respond like this. And I think this is really great. We don't need maybe to replicate completely our research in both sexes, or kind of taking everything into account, but really getting some help from these computational models, machine learning, and even AI maybe. So this kind of come to my conclusions, where I hope I answer my my starting question. So are sex and gender important in basic science? I think they are. They are because they can increase reproducibility and efficiency of our research quite a bit. They create a lot of new opportunities for discovery. I think this is, as a scientist, should be really exciting for all of us. It can improve outcomes, and for not only for female, but I think also for the entire population. And it will reduce translational crisis, so this crisis that we are seeing when there is a lot of things that we find out at the basic science level, but then we go into preclinical and clinical studies and things don't work out. And maybe that this is because we're not looking at the right thing at the beginning. And I think also, this is more a philosophical point, I think if we already at the basic science level are aware of the sex of our subject of investigation, this will kind of make the other levels of research, the preclinical and clinical research, also more aware. They have to be, because I, as a basic scientist, I was already, interested, and I was already looking into that. So with that, I kind of would like to thank the people that are funding my research, especially the DZHK that is funding cardiac sex difference lab, and so you can see the lab trying to jump. And I also would like to thank Eleonora Grandi, who is this computational modelist that I work with at the University of California, Davis, and as well a couple of my mentors that have been really great promoters of my research, and also sex differences in cardiovascular research, especially Leslie Lineman, who is a pioneer on this, and Mitch Gotthard from the MDC. So thank you very much. That's it.
Are there questions for Dr. Crocini? If not, there will be plenty of time to ask questions later in case you have them.
Thank you.
And we will go on with our next speaker. Our next speaker is Professor Dr. Sabine Klaassen, and she heads a research group called Clinical Cardiogenetics at the Experimental and Clinical Research Center of the Charité and the Max Delbruck Center. She's also a physician, and she's specialized in cardiomyopathies and genetics at the Clinic for Congenital Heart Defects and Pediatric Cardiology of the German Heart Center of the Charité. And today, Professor Klaassen will talk about a study that has been very recently published, I think mid-October, she told me it was published, and it's about one specific mutation that determines sex-specific cardiac nutrient metabolism.
Yeah, thank you very much for the introduction. Actually, I will present an example of a model, a human model, and the correlating animal model where actually sex matters, and so I'll get right into it. So my interest has been in human genetics and identifying the genes that cause cardiovascular phenotypes, certain cardiovascular diseases, especially cardiomyopathies. With this effort, we noticed one syndrome that you find up here, which is called chromosome 1p36 deletion syndrome, which has different features. It's the most common human terminal deletion syndrome. It has craniofacial dysmorphisms, it has neurological features and problems in muscle and skeletal system, and also cardiovascular features. And these are congenital heart defects, so anatomical heart defects, but also cardiomyopathy in a certain percentage of cases. So what are cardiomyopathies? These are heart muscle diseases. And up here, I don't know if the pointer or the mouse is not working. So on the left side, you see a phenotype of dilated cardiomyopathy. And on the right side, left ventricular non-compaction. And actually, you can see here the ballooned left ventricle, the left chamber of the heart. And on the right side, you see this non-compaction phenotype. It is the right ventricle and the left ventricle. So both chambers of the heart are malformed. They have a spongy appearance. So this goes together with arrhythmias and also heart insufficiency. So this is now, this is all a bit, the lineup is not correct here of the text. So, but I just gone. So we identified a disease gene which is called PRD16 already 10 years ago, and this is one project we are working on. And we continue to work on this and with an animal model that I'll show you later. So just because this is important at this point, when we identified this gene in the syndromic patients, all patients that have this syndrome and have cardiomyopathy are shown here in yellow. A certain gene is deleted, and that is PRDM16. And we identified 18 individuals, and in 16 of these 18 individuals, they were females and only 2 were males. So obviously, there's a sex bias in this syndrome. And there are also non-syndromic forms of this cardiomyopathy where we screened and found mutations in this gene as well. It's a zinc finger protein, it's a transcription factor, and it probably leads to haploinsufficiencies. So if the amount of the protein is reduced, then you run into these problems into the phenotype. So what was known? Nothing was known in the heart about PRDM16, but it is very crucial in the cell fate determination of myoblasts into brown adipocytes. And brown adipocytes, they get their brown color from the large amount of mitochondria they contain, and they are very important in thermogenesis and also for now the treatments being sought for brown adipocytes to treat diabetes and obesity, as it's an important this brown adipose tissue is important for like an endocrine organ, and it's important for systemic energy metabolism. So the role for PRDM16 in cardiac development and cardiomyocyte function was what we tried to look at. And DCM, dilated cardiomyopathy, there are very many genes. If they are altered, lead to this phenotype of dilated cardiomyopathy. And up to now, the evidence that PRDM16 is really a cardiomyopathy gene was limited. We had a zebrafish model that we already published, and now we wanted to go further with animal models. One model had already been published. That's a biallelic conditional knockout of PRDM16. This is a very late phenotype that induced severe cardiac dysfunction and hypertrophy. And also already there, mitochondrial dysfunction could be shown. So we took another approach. We used a model with an early we wanted to look at an early pathological window and a systemic mutation, because in the patients also it's not conditional, but it's systemic germline mutation. And from what we knew about the syndrome, where we had the sex bias already, and from the function of PRDM16 in brown adipocytes, its metabolic function, we right away divided the animals into male and females. And we used this model here on the left side, which is an Inu mutant, and the homozygous mutants cease shortly after birth, and the heterozygous ones had not been investigated. And this is where we started. And these on the right side, you see the results. We saw reduced body weight in these animals. We saw the relative left ventricular weight was reduced, especially in females. Females are shown in red and males are shown in blue. And on the lower panel, you see that the cardiac performance was reduced, stroke volume, cardiac output, and we see that females are more affected. And we went on to do transcriptome analysis, where we saw moderate changes. And we see here that on the plots for males and for females, and the females are more dysregulated on the right upper panel. And then we see here the females plotted against the males on the lower panel. And we see that PRDM16 itself is upregulated, which suggests an autoregulatory control of transcription. We identified two factors that are really regulated, which I will not go into in more detail at this moment, pyroxD2 and PBZIP1. And the function of these is not known. Proteomics also showed that these factors were upregulated. So we noticed a reduction of the central metabolites in amino acid, glycolysis, glycerol, and TCA cycle for both sexes, actually. And then we went on to look at the cardiac nutrient metabolism. This is just and a few examples. Much more was done. We could show that ATP turnover was increased and that oxidative capacity was reduced only in female PRDM16 heterozygous knockouts. So with in silico analysis, actually cardiokin, a model that was developed here in Berlin, we could look at the cardiac tissue, and this revealed that the maximum fatty acid utilization was decreased in males, here on the lower panel left, and in contrast, female PRDM16 mice had a diminished maximal glucose utilization, while the males were unaffected. So in summary, our studies suggest that metabolic dysregulation is an early event of the PRDM16 associated cardiac pathology. And we see that cardiac nutrient metabolism is affected in a sex-specific manner. Female mice develop a more pronounced phenotype, and this indicates sexual dimorphism. And obviously, female PRDM16 mice compensate for disturbances in cardiac metabolism less efficiently than males, and this was just published. And my conclusions and outlook are that cardiomyopathy phenotype must be checked for sexual dimorphism at any pathological stage, and that that actually the sex differences in metabolism might drive the severity of other cardiovascular phenotypes as well. So mostly the experimental designs that we have today, as already mentioned by my previous speakers, have not addressed the issue of sex differences. And in the future, I and already my other speakers, I think we would suggest that in vivo and in vitro experiments in mouse, but also in induced pluripotent stem cells that we are working with, should include both sexes in our research. So I thank you for your attention, and I'm happy to take questions now or later.
Are there any questions for Professor Klaassen?
One brief question. Thank you first for an interesting talk. With this mutation, when you find 18 persons having a syndrome, 16 of those are women, to my mind, that could either mean that male fetuses with the mutation spontaneously abort or they die in childhood, or it could be that they just don't develop the disease without knowing sort of the background distribution in the population. Do you have any thoughts on that?
Yeah, that's a good idea. At this point, we didn't know. But now, since we got the animal model, which really shows the sex differences, we think that's the case, that actually they are more severely affected. Actually, the PRDM16 mutation doesn't cause very severe cardiomyopathy. It's a very subtle, the heterozygous phenotype is a very subtle phenotype. So we rather think that it's the sex bias and not the other.
Okay, I think we're going to move on to the next speaker because we're also a bit short in time. So the next speaker is Dr. Hanna Hörnberg, and she's a neuroscientist who leads the Molecular and Cellular Mechanisms of Behavior group at the Max Delbruck Center. And her group is interested in understanding the biology underlying social and emotional behaviors in health and disease. And we already saw from this previous speaker how important it is to include both sexes when studying disease. And also in her lab, researchers use both male and female mice. And also they have ongoing projects that specifically focus on identifying sex-specific factors linked to symptom development in autism and depression. The floor is yours.
Thank you. So thank you for that kind introduction. I was very excited to be invited to participate in this really great session. So I started my group at the Max Delbruck Center for Molecular Medicine at the Buff campus about 3 years ago. And in the group, we're interested in studying mental health conditions. And we're studying these using mouse models. So I will really only talk about sex today, since it's not really possible to study gender in animal models. So mental health conditions, which according to the WHO definition includes everything from autism to eating disorders to depression. They're relatively common, so about 1 out of 8 people in the world will be diagnosed with mental health conditions during their lifetime. And the most common ones are major depressive disorders and anxiety disorders. And they're marked in red here on this graph from Lancet Psychiatry. And as you can see on the shift of this graph to the female side of the right, both these conditions are more commonly diagnosed in women compared to men. And in fact, pretty much all conditions that affect the brain have a sex bias in diagnosis. So as an example, I was sad to know that Alzheimer's disease is more commonly diagnosed in women versus Parkinson's disease is more commonly diagnosed in men. And as Sofia has already, sorry, I don't know why it's a gray part of the screen. It's not supposed to be there, so ignore it. So there can be lots of different factors, both kind of lived experience and biological factors, why we see this sex differences in diagnosis, and Sofia has already very nicely covered them, so I will not go through it. But the point I really want to make here is that we don't actually know for any of these conditions why we see a sex difference in diagnosis. And one of the reasons we don't know this is because we have not, as we already heard today, been particularly good at studying this. So despite this very clear sex and gender difference in diagnosis for mental health conditions, males are still very much the norm in clinical and especially preclinical research. So the graph I'm showing you here is from my fields of neuroscience, and it's the percentage of articles who broken down by which sex they use from 2009 to 2019. So this is mostly preclinical research or using animal models. And females are in orange, and as you can see, this is still very small, so it's less than 9% of all articles. And although the number of articles that use both sexes, and again also this unspecified, which is really unhelpful, but the number of articles that uses both sexes have really improved, which you might think then is a good thing. But the number of these articles that actually do any type of sex analysis has not changed and is still less than 25%. So this increase in articles that use both sexes have not actually led to any kind of substantial increased knowledge of female biology. So why does this matter then? So it matters because we know that neglecting sex differences in clinical and preclinical research has real-life negative health consequence. So as an example, it's well known that men and women can differ in their drug absorption rate and metabolism, and this can affect how well a drug will work for a person. And because most drugs are calculated based on men's body weights and metabolism, we think that because of a result for this, then women are much more likely to experience adverse side effects of any drugs they take, including really serious side effects that requires hospitalization. And that's already been touched on a little bit today by Sofia. We also really don't know how sex and gender specific medical factors like hormonal contraceptive or the use of sex hormone impact things like mental health conditions like depression, or how they might impact treatment response. So I hope this first part have really convinced you that to give kind of the right treatment to the right person and improve treatment response by also making sure we have as few side effects as possible, it's really important to consider sex and gender specific factors when we do our research. But having said that, it's also really important, I think, to keep in mind that sex is really not everything. So I think maybe instead of just looking for sex differences all the time, one way to do it instead is to just focus on using a population that's representative of whatever it is you're studying. So for all the studies in WeLab, we always use both sexes since this is representative for what we're studying. And if we don't see as sex differences, we continue to use both sexes for all our other analysis. We're still interested in things like individual variability because we still see this in mouse models, so we can still kind of use this then to look for more personalized medicine approaches. But if we see a sex difference, then we will split the groups, still keeping individual variability in mind, but then also trying to understand what part of the male and female biology might be responsible for this sex difference. And in most of our studies, the first part is what happens, so we actually don't see a sex difference that often. But I will tell you very briefly about two studies we've done in the lab where we did see a sex difference, and where if we had not studied both sexes from the start, we would have really missed some important biology. So the first example is from a really short study we did looking at the molecular mechanisms in autism. So autism, which is a condition and not a disorder, it's diagnosed up to 4 times more commonly in boys, a bit different depending on the country. And there can be lots of different reasons for this, including like just a diagnosis bias, that doctors are really bad at identifying autism in women. It might also be that we expect really different things from boys and girls, which leads to this diagnosis bias. But there's also been a lot of research, or at least some research looking into the fact if there is some kind of biological factors in the male or female biology that leads to boys more commonly being autistic compared to girls. And in the lab, we're interested in the synapse, which is the connection between neurons that allow them to communicate and send messages, and where things like our memories and experience are stored. And for this project, we were interested to see how genes that are associated with autism, if we remove these genes, how this affect the synaptic function and how synapses can communicate with each other. So for this project, what we did is that we took male and female mice. So these here are wild-type mice that have no changes in their genome. And then we isolated synapses from the brain. And then we use a technique called proteomics, which allows us to look at thousands of proteins in these synapses. And for this project, we use a specific part of proteomics called phosphoproteomics that allows us to look at the phosphorylation state of all proteins, which is kind of like the activity level of the proteins. And the data I'm showing you here is in the form of principal component analysis, and how these plots work is that the further away the dots are from each other, the more different they are. So the closer they are, the more similar. And as you can see, the male and female dots are completely intermixed, suggesting then that sex has no impact on the activity level of synaptic proteins in the wild-type condition. So we did the same experiment in a mouse model that lacks a gene called neuroligin 3, which is a synaptic protein that's been associated with autism. And if we do the same experiment, then all of a sudden we see a very good separation between the sexes, suggesting that the biology of the sex-specific biology of the mice kind of matters for this mutation. And if we look at why this is, It's because the mutation doesn't really seem to do anything in female mice, at least on this particular measurement. So these plots here are called volcano plots, and the dots marked in red are proteins that are where the activity levels of the protein is significantly changed between the mouse model and the control situation. And the female plot is to the left, and there's no red proteins there, whereas the male plot is to the right, where you have lots of red proteins. So there's lots of things changed in the males and nothing in the females. And if we look at what these proteins are, they're primarily proteins that affect how the synapses work and how they communicate. So if you would have done this study only in males, which is then the most common, and then try to apply this to both male and females, it might not have had any effect or might even have been harmful. And if we would have done this only in females, we would have come to the conclusion that this mutation doesn't affect the signaling pathways of synapses, which also clearly would have been wrong. So not using both sexes would have left out some really crucial information that's really important to try to understand what this gene does at the synapse. So another example I want to give you is from a project where we really from the onset decided that we wanted to look at sex differences. And this is a project where we looked at sex differences in depression. So as I already mentioned, depression is more commonly diagnosed in women compared to men. But the symptoms of depression also differs on a group level between sexes. So on an individual level, this is not necessarily meaningless, but on a group level, we can see differences. So for example, women are more likely to experience changes in appetite or depressed mood, and men are more likely to experience anger and substance abuse issues. There are lots of risk factors for depression. But the one we focus on is one that's known to be a quite strong risk factor, and that's to experience a traumatic or stressful event, especially during childhood and adolescence. But we also know that most individuals that experience something traumatic will not go on to develop depression later on in life. So most individuals will be fine. And we call this individual resilient. And then some individuals will go on to develop depression, and we call these individuals susceptible or vulnerable. And for this study, we wanted to know, try to understand why some individuals are more vulnerable to depression or to develop depression after stress than others, and if sex as a biological factor has any impact on the vulnerability to stress. So obviously, how can we study depression in mouse models? So mice are really not humans, and depression might seem like a very human condition. And how it's diagnosed is that you need to have 5 or more symptoms of depression for at least 2 weeks. And some of these things we obviously cannot model in mice, so we can't ask them if they feel worthless today or not. But a lot of the other things we can model. So for example, we can look at anhedonia, or the ability to experience pleasure by seeing how much the mice are interested to eating really tasty food, which is a little chocolate cereal that they really like, or how much they like to drink sugar water, which they also really like. We can look at their energy level or sleep by just studying them in their home cage without disturbing them at all and see when they sleep, how much they move. We can test their memory by looking at how well they can discriminate between different objects. And we can also directly measure their stress hormones to see their kind of physiological changes to stress by just doing a blood test. So if we exposed male and female mice to a very mild stressor during their teenage years, and then did all these different tests for more than 2 weeks of time to see which mice were susceptible to develop depressive-like phenotypes after this, we find that in both males and females, about 50% of mice would show a depressive-like phenotype. So it doesn't seem then that sex has any impact on the vulnerability to stress. But since I'm telling you this is obviously not the whole story. So again, what I'm showing you here is a principal component analysis, and this is of all tests we did in the animals. And female stressed mice are here in pink, and the male stressed mice are in light blue, and the control mice are in the middle. And as you can see, the male and females stressed mice are moving in kind of opposite direction from the middle. And this tells us then that stress seems to really affect male and female mice very differently. And if we look at why this is, we find that male mice, they have what we would kind of call a classic depressive-like behavior or phenotype in mice. So they don't like sugar water as much, they have poorer memory, they have higher levels of stress hormones, and they also have changes in their sleep and activity. The female mice, though, they don't have that many behavioral changes, actually almost nothing at all. What they do show is clear physiological signs of stress. So they have changes in their weight and increased level of stress hormones. And even in things they have in common, like changes in sleep and activity, this is also different. So male mice are less active and, if anything, sleeps a bit more. And female mice are more active and sleep a bit less. So if we had done this study only in male mice, for example, then and try to apply whatever we found to both sexes, this might also really not have been helpful. And we're now trying to understand why we see these big sex differences and see if we can use this to try to then develop kind of better treatment approaches for depression. So with that, I want to leave you with two take-home messages. So one is that I hope from all of our talks today that you will agree that it's really essential to consider sex as a biological factor when studying the brain and brain disorders. But the other thing then is that sex is really not everything, and on an individual level, sex differences are often meaningless. So even if we can see differences in most things we look at in the brain on a group level, it doesn't mean anything for you as a person. So instead of focusing on sex differences, I think we can start focusing on using a representative population of whatever we study, no matter what that is, in all ways we can make it representative, so that we can move from a one-size-fits-all to a more kind of personal medicine approach that, of course, then has to take both sex and gender into account. And I first, before ending, really want to thank the fantastic team who is doing all this work. So specifically single out, so I can't point at them now, but Maria Steinecker, which is kind of in the middle, so because everyone wears the same color, so it's not so helpful. She is the one who did most of the work for the Synaptic project, versus Tobias Poole, who you can see because he's the only guy. He's the one who did most of the work for the Stress project. And yeah, thank you to all of you for your attention and for coming here today, and I'm happy to take questions now or later.
Are there any questions for Dr. Hörnberg?
Thank you very much for this fascinating talk. I was really impressed by the data that you show. I have one question. Could the differences that you see in the females, that they don't show this behavior or this obvious behavior change, be related to females have to give birth and take care of kids, so that they're more stress resistant, don't show it that much?
Yeah, so that's a very good question. So in this study, we looked at adolescent mice, so they're quite young. So we did the stressor during the time where their period starts, basically. So we would think that anything or that factors like that might have less of a role. But this is something we really have to keep in mind also looking at animal models, that they might have really sex-specific factors that are important for their reproduction health and how they normally live their life, then we can not at all translate to humans. Which is why we really have to look kind of at their brain and try to understand what this difference is. Since they physiologically are quite similar but behaviorally quite different. It might also be that this stressor affects them differently. So this is a social stressor. So basically we ask the mice, or we have the mice move house very often. So they constantly meet new mice. And this is a bit stressful for them, like maybe it would be for all of us if we constantly had to live with new roommates every 2 days. But this might be more stressful for males than females also, which might be why we see a stronger effect.
All right. For time reasons, I want to maybe ask you to ask your question later during the drinks. Thanks for that. Then we can move on to our next speaker and also the last speaker. And that is Gertraud Turu Stadler. And she holds a professorship for gender-sensitive prevention research and heads the Department of Gender Research in Medicine at the Charité. She's also a member of the Steering Committee Diversity and Gender Equality of the Berlin University Alliance. And she is an advocate of gender and diversity-sensitive medicine. And her research mainly focuses on personalized, digitally enabled disease prevention. And today she will talk about methods to systematically assess different dimensions of diversity in studies.
Thanks so much for having me here. So it's a pleasure to talk after all these very exciting talks that we just heard. Yeah, so I'll talk today about closing the gender and diversity data gaps in the health sciences and what we all can do to contribute to that. So definitely the previous talks have addressed that too, but like I want to maybe make some suggestions what we can do in research with humans mostly. So of course there would be nothing to report without my team. Like so they are very crucial to this endeavor, and I want to thank them up front. We have gotten seed funding from the Berlin University Alliance to tackle these things. So what is the problem? Well, we have massive gender diversity data gaps and throughout the health sciences. Yeah? So in the US, after several tests were excluding women systematically from research due to fears that there would be damages to the embryo. So women were systematically excluded from health science, yeah, so from randomized controlled trials. So mostly these trials were then run in the initial ones in men, and then in young healthy men, of course, to test for toxicity. But then also the additional trials were mostly done in male populations. And in the US in 1993, there was a law really demanding the inclusion of women and ethnic minority participants in all studies. Now, this has not happened in Europe until much, much later. So we have a lot more data from the US and North America and Canada and Australia and so on that really systematically include, tell us something about gender and diversity. Because, So some diversity domains are routinely assessed and measures are able to cover the spectrum of characteristics, but individuals who are situated at the margins are not included. For example, the prime example is age. So very young participants or people and very old people with many more comorbidities are routinely excluded from studies. So we know not that much, so we have data gaps there. Then some diversity domains are routinely assessed, but we lack inclusive measures that adequately capture the spectrum of individual identities, for example, for gender. Yeah, so gender minorities are also, so we have a very binary conceptualization of sex and gender usually in our studies, which really, and Sofia has really made a very good case. We need to do better there. And then some diversity domains are hardly assessed at all because they are taboo or they are not considered interesting. Like race is considered taboo, for example, religion as well, interestingly, in Germany. And then care responsibilities seem to have not been considered important enough to include. So we think that this needs to change, yeah. So and that this cannot just be the field for us enthusiasts. Of course, we need to spearhead this, but we need to, in a way, do this in a systematic way. And when I started 4 years ago at the Charité, I met all these nice colleagues, and they were like, "Oh, Turu, I'm really interested in diversity and gender questions, but my questionnaire is already too long." Yeah. So and then don't you have something really brief and easy that I could just take off the shelf and use it? Well, and then I was looking, and I was like, well, no, not really. So we don't have that, like a standard short measure that we can simply use. Now, we put together first ideas for a solution, and here's our first take for this. So together with about 54 experts from the Berlin research area, but also international experts, we put together a diversity minimal item set, the DIMIS, to foster diversified innovations in the health sciences. I couldn't agree more with what my the previous talks, that not always does gender and sex matter massively, but for many areas we really don't know. Yeah? So we do need to understand it better. Where does it matter and where does it not matter? And the same, I would say, would be applicable to the other diversity domains. So which ones are the ones that we thought would be really interesting? Of course, gender and sex would be a good starting point. And I think if we are not completely sure that sex and gender do not matter, then we should assess them. So then, and it is often assessed, but in a very binary way, and we need to change that. Then age is routinely assessed in health sciences, and sometimes also socioeconomic status, like things like education, neighborhood SES, and so on. But then, so we have another bunch of diversity domains that we thought would be really important to routinely assess, which is care work, and then a whole health comorbidities like mental health, physical health, and disabilities. And then we have our problem sector here, which is sexual identity, ethnic and racial identity, and religion and worldview, which are considered kind of like too personal and you shouldn't ask them in routine studies. Well, so we have had a lot of experiences with this, and I would kind of politely disagree wherever possible. I would try to include them. But okay, I do understand that questionnaires need to be short, and these diversity domains may not matter for all research questions. By all means, do adapt them, but give this some thought. So what are our ideas, what we could do with these? So, of course, the first is that we can use it for research, to improve the sample descriptions and discuss the generalizability of our research findings. So if I in my studies, mostly use patients that have only a single chronic disease, for example, a heart disease, but do not have comorbidities, then the medications that I will develop will probably, so we need to check then in more health services research, how do these medications do for people with multiple chronic conditions and multimorbidities. So this is very important to really describe what do our research to whom do our research findings even apply. Then, of course, we need to also use that to pool data to detect smaller effect sizes for single diversity domains, but also to reach critical sample sizes for smaller marginalized groups, and also, of course, for doing intersection analysis, because, of course, not all women and men are the same, and we would really like to look at the intersections with other diversity domains. To develop personalized and innovative interventions that are tailored to the needs of diverse populations. So what we really want to have is personalized medicine, and we are tailoring to all kinds of genetic conditions. But in a way, why don't we do the simple stuff that is like really tailor to the more superficial diverse domains too? So definitely a very useful tool to use for research, and I would love to see this used much more and done more systematically, because otherwise in our meta-analysis, we are still always starting on square one and really cannot harmonize our data. Now, there's also, and I thought this was very interesting, the idea to really use this in clinical practice. And there we are not very far, but at the moment we are trying to implement this with the hospital information system here to allow patients to self-identify on gender and diversity domains that matter for the healthcare. And of course, simply, it is a matter of politeness to ask people how they want to be addressed, I think. Yeah. So, and I think we should do this better and more systematically. We also discovered that, for example, the Charité system, everybody seems to be a native German speaker and basically of German nationality. Well, it seems that this is a default setting that cannot be changed, and it's really a bug. Yeah. So it's unbelievable. So in clinical practice, we need to do much better. We have no translation services for people who do not happen to speak German or English, and this is really scandalous, and we need to change it. Now, a third use of this is for policy development and policy impact analysis. Well, let's take a quick look back at what happened in Corona times. Yeah, so COVID-19, there were many policies developed, but there was no systematic way of really doing a balanced, basically checking if the policies had balanced costs and benefits for all, and really analyze the impact of these policies on diverse population subgroups. So, for example, for people with care work, there were really catastrophic effects of some of these policies, and they were kind of like really not taken very seriously. So I think in the future we can do better, but so I discussed this already with our esteemed colleagues at the Ministry of Health, and it was quite interesting. So they do not have systematic tools to do this. Yeah. So I would love to see a more systematic use there. And then, of course, we are also aware that different people do ask different questions, and diverse teams have more innovative research outcomes. So it would be really interesting to use these diversity tool sets to describe the lived experiences and diversity domains covered in on the organizational levels, and here not on an individual level, to safeguard personal privacy and avoid forced outings and exposure, but rather to see, do we have leaky pipelines? Yeah, so who makes it to the top or even progresses and can stays in an organization, and who gets kind of either excluded or drops out. And so at the moment we know for women that kind of the higher we go, we have less women, but otherwise we don't have this data. So we cannot actually at this moment do any evidence-based organizational development because we don't have good baseline data that show us like how do our pipelines work and who is can unfold their talents and who does not. Anecdotally, of course, we know what's happening, but yeah. So how did we do this? So we assembled as diverse a team of researchers that I could get my hands on. We did an extensive literature search. We did semi-structured interviews with scientific experts and community stakeholders, then held several workshops with diversity researchers, consolidated the feedback, and then put a diversity set together. So it's published, and the goals were to be inclusive and non-discriminating, brief, that we can compare with general population data, so that we can really describe, so how does our sample diverge from the whole population, and it's available in English and German in the published version, and at the moment we have, I think, 26 languages in the works, so it's very nice to. If anybody is interested and has a heart and talent for a certain country, then please contact me, because we are trying to have as many languages available as possible. So what did we find? So, for example, for asking people, so assessing gender and sex. So the recommendation would always be to consider the order in which you are asking these. Which answer options you give. So for example, we have to balance out that we have options available for gender minorities, but that also people who are not used to these, to dealing with gender minorities, that they still find what options apply to them. So one possible solution for a not very woke population, for example, elderly patients, maybe, and this may be a cliche, I wouldn't want to be mindful about that, but so to start with things that most of them will be familiar, like female and male, and then add additional identifications such as inter, trans, non-binary, and so on. And always include a prefer not to answer option, and also allow for self-identification, so that there's always a way that we can learn more about how people want to be self-identified. Then we are trying to avoid othering. So we always say like, I prefer to self-identify as instead of othering, and then clearly explain why the data is being collected. And now this is very important. This data is really sensitive, yeah. But we deal with sensitive data all the time. We have genetic data, we have stigmatized health conditions. So we are used to this, so we can do it. But we definitely need to guard this data with our life, yeah. So we need to really be sure that we can that this data is not used against minoritized populations. Now I've brought a few study examples. So here, evidence-based organizational development. So we are part of the Berlin University Alliance at the Charité. So does the Charité represent the diversity of Berlin? So we did the Charité Stärken survey with students until the beginning of this year, and then now have a representative sample of the whole Charité staff members. And so thanks everyone who participated. So we are currently, so that just ended in the end of October, so we are currently analyzing, but I brought a few tidbits. So the idea is here to do a needs assessment, and then do really focused intervention development and testing with our junior research groups to try to develop targeted interventions for plugging these leaky pipelines for our talents. So what did we find for students? So we have a little bit of an overrepresentation of women. So we have about 66% female students in medicine. So women are always more interested in these topics, interestingly, and so we do have but we can kind of weigh, so to really make the sample representative. Very interesting to us was that 2.5% of our students say that they are gender incongruent, and so that was much higher than we expected, and we do not really take that into account so far. what was kind of very interesting was that there's a lot of invisible diversity that we are not having any services for at the moment. So we know that we have, the Charité is a really hard place to navigate if you have a physical disability. But so we knew that, and about 3% of our students identified as living with a disability. But we also found that about a third of our students said that they have a diagnosed mental health condition, and another third said that they have a chronic health condition, a physical health condition. About 14% say that they do care work, and what also was very interesting, about 1 in 4 said that they are first-generation academics, which we didn't know at all. And here, this was also interesting, like, so about a third of our students say that they have a history of migration in their own generation or in their parents' generation, and that the ethnicity they identify with is so most of them identify as white, but there is a large spread of other identifications, and I think this measure wasn't optimal. Yeah, so we need to improve that. It was probably too fine-grained, but but at least what we could show is that so we can measure ethnicity, and and not too many people say I wouldn't give you that data. Yeah? Here we have now, so this was all done with Pichit Puspavanich and his junior research group here at the Charité in my team. And then, the next one is Marina Fischer. And so she did a survey of young creatives, a nationwide study of art school students. And so here is the model that we are testing with Marina Fischer's setup. So it's kind of like trying to understand what Me Too does for young creatives. And so we were shocked to find how many microaggressions, abuse of power, sexual harassment, sexual aggression, and technology-facilitated sexual violence we found, like relatively early on, like experiences of hostile behavior within, but also outside their art schools. And these are publicly funded art schools. So we used the diversity domains to understand, like, so how are they related to mental health and well-being? So depressive symptoms, well-being, professional thriving, and feelings of closeness and belonging to the creative industries. And then understanding, so how do these hostile behaviors affect that link? So here's a very interesting finding that surprised us. So we found that, so women have, so how do we analyze this? So we were looking at a Flinter conceptualization of gender, so women and female and female identified people, and have more depressive symptoms, and then so this was actually mediated by microaggressions. Yeah, so this is of course a cross-sectional study, and there's lots to be done. Ideally, intervention programs to help with both bystander interventions and lowering these hostile behaviors directly, but also mental health interventions. Yeah, so definitely, so there seems to be a case that so people with diverse backgrounds have worse mental health, and so some of this is going through this pathway of more severe discrimination experiences and hostile behaviors. Now I'm bringing you like the last example here, and so here is an example of a personalized digital intervention that we ran in the US. So this is our preliminary study, but now we've done the full randomized controlled trial that we are just got in press, I think, but I didn't manage to put it in here. So we have the problem of high alcohol use, and we're testing personalized text messages versus just doing a weekly assessment of alcohol use. And what we are seeing is that these people are very high in their alcohol consumption, like 24 drinks per week at baseline here. So very, very, very high. So it should be rather like more 4 drinks or something like that, right, per week. So this is very, very risky drinking already. So in the control group, we find that not much changes by just monitoring your own drinking weekly. But we found that personalized messages that are tailored adaptively perform the best in bringing down this very risky drinking. And you got to keep in mind, these would be people who would never seek any treatment just yet. Yeah? So they are secretly drinking. They know that they have maybe a bit of a problem. But and in a way, so we are hoping to personalize much more. So by collecting more data, really running very large-scale trials, and so I hope that soon we'll have like really personalized medicine. So thanks so much for the attention, and now I'm looking forward to our discussion.
So I would like the audience, if you have any questions for Professor Stadler, please find her after the event, because then we have some time to still discuss during the panel discussion. So I would like to invite all the speakers to the stage. A large fear when organizing a panel of most organizers is that they end up with a manel, so basically a group of only men. And as you can see here, we don't have any. And I'm wondering, because this is basically maybe actually worrying, because I would say, is gender personalized medicine something that only women should be interested in, or are we the only ones benefiting from this? I'm wondering what you think about this. Are there others that also benefit from personalized gender medicine?
Well, I would very much dispute that. Yeah, so men live in most OECD countries on average 5 years shorter than women, and so there is definitely a lot to be gained for understanding these gender differences, and really for everyone. And so, to me, it's kind of a bit of an awkward thing. Like so, and somehow there seems to be a blind spot in the whole field that gendered innovations is about women's issues. Yeah. So, and I think we really need to do better at that, and in a way spread the word, because that's really not what's going on. Yeah. So there are a lot of conditions where the cascade is actually like really damaging to men. Yeah. So and has a disadvantaging for men, and so we just need to work on that. I think so. It's mostly, I think, an awareness problem, and also I think everybody. So we won't plug this gender and diversity data gaps with just a few studies. Yeah? So we need to really systematically look, and so we need all kinds of male non-binary allies. Please help us.
Any other comments on this topic?
Just to add that this is also strategic funding-wise, even though we do have, as I mentioned before, pressure from funding bodies and research planners to do things about this, I still think that by pointing out what you just said, we might perhaps get even better support for it.
I also would like to add that, so I agree with all of this, and to get personalized medicine, we have to study everyone, otherwise it will never work. It's not black and white, so there's a huge Venn diagram between sex differences. But also, I think it's fine if it only benefits women. Not everything has to benefit everyone. It's like fine to do something that just benefit one smaller group as well. And women, it's not even a small group. It's 50%. So
I think we made very clear through all of your talks what the problem is. We identified, I think, that very well. And I think, Professor Stadler, you also mentioned what scientists can do here. But I think there's also other stakeholders in the field. I'm wondering what other people should be asked to the table in order to really bridge this knowledge gap.
So yeah, I mentioned in my presentation a little bit that we need also the rest of the system to kind of be even aware of sex and gender. So I know that now funding agencies require at least a justification on why you're not going to use both sexes if you're working with animals, but it's still like you can always work around it somehow. It's not. I mean, I don't feel that it's completely in place right now. So I think a lot has to be done at the level of where the money comes, because only by changing how we fund research and really where money goes, we can really change the thing. So this is, I think, the most important, in my opinion, part. Yeah.
So
so the funders, the funding organizations have a really big role to play there. And in a way, good practices are to really make it mandatory in all applications to report. Like, so is sex, gender, diversity analysis relevant for your research question? And if it's and you kind of like have to argue, and the reviewers need training to recognize, is this a good answer or not? So so definitely funding organizations, reviewer training, but also then to so, for example, the Canadians have this really interesting clause that any publicly funded research needs to report gender disaggregated results for their main outcomes. And I think in the US we had since the 1993 this law to include women and ethnic minorities proportionally in the studies. And so as they are represented in the patient population, and also report your results that way. So we don't have that. So there is there's really something to be done there. And then so editors and journal reviewers we can do something there, so to train them. And so a lot of journals now require a much more thorough sample description. I think that's a very good thing.
I want to comment onwards on that from editors and journal reviewers to the sort of more wider field. There's a saying from, I think, originally of a disability movement, nothing about us without us, which is a call for participation in research, ideally at levels of study design and carrying out research and interpreting research, but at the very least in the presentation and interpretation of the findings and the framing of the findings that I think we are missing. And I think this holds for many different demographics that are in a position of power imbalance. It holds because there's so much research that's been published still today that is sexist in its interpretation. There's research that disparages the abilities of women. There's research that disparages the abilities of non-white population. There's a lot of research published in putatively peer-reviewed journal that is slandering LGBT people in a variety of ways, misinterpreting things. Sometimes this is malicious, but many times I think this comes from an inability to understand what it is that one's study instruments were actually measuring, which really underscores the necessity, crucial value of the research you were presenting, Turu, in that if you're just looking at something through a very narrow lens, you may see part of the elephant, but you will not actually see what you're looking at. And having at least some representation at any stage in research, I think, would go a long way from preventing, like, newsflash, discoveries, this population is stupid, from being published and brought along the Twitter sphere or whatever. And I would really love for scientific journals to come together in a sort of code of conduct to demand this in the review process. I think this is something we could and should do. That is one aspect of stakeholders in diversity research more general, but definitely also in the more specific case of sex and gender.
Yeah, following up on that a little bit, it's not, I mean, in some cases it definitely is, but it's not often that scientists are kind of evil. We're all under a lot of financial constraint and publish in pairs constraint, and using both sexes in a lot of cases, then it takes double the amount of time. And we're not necessarily given that time or the financial resources to do that. So you really need kind of a shift in the entire scientific funding and how university rewards things in order to really enable us to do this research in the best possible way, because it will take more time and it will cost more money, but it's obviously worth it. So yeah, you need everyone to get on board, I think, to make this happen.
And if we expand basically the realm of diversity from just sex and gender a bit broader, because during this Berlin Science Week there has been a lot of programs about diversity in science. I'm just wondering, basically, also what you are referring to, how important it is to have a dialogue with the group you're researching, for example. I'm wondering what your opinion is on hiring a diverse research team. Do you have any suggestions on how to make sure that the research is not just done by a very homogeneous group of people?
Well, we do have some first experiences with that, and one issue is like so. It's not easy. Yeah. So when I go to the so we do have a very leaky pipeline starting really from childhood in Germany. So when it comes to ethnicity, I go into Berlin schools and they're full of black and brown faces, and these people are not represented in our student body. So and in a way but really to say we are trying to really become much more welcoming and really reach out and work with schools to encourage them to enter our medicine and enter health sciences and really feel at home there. So we find as well that kind of anybody who's non-traditional, first generation academics and so on, so they everybody when they come to university or to this new situation, like your little rats, who it's a little bit of a stressful situation. Yeah. So but for people who are more privileged, so this feeling of not belonging subsides relatively quickly, while for others it never stops. And this is really this feeling of not belonging, we can all do something to create a more welcoming environment. So non-traditional, underrepresented groups drop out at much higher rate when we manage to attract them. And so we can do much better there, I think. So, and then of course, within teams, there is lots of things that we can do. So I'm a first generation academic myself. I was mentored by some first-generation academics, also by some very privileged people, and so there's different things that different people can bring, and so we need all talents here, and so to really work together to create this welcoming environment is quite a challenge, but I think it can work.
I think a lot we can do also in the narrative that we give, that we describe research, because there is this obsession that we are doing this just for passion, and I feel like passion is like, I mean, what kind of privileged person can just, do their thing for a passion? Like, we do this work, of course we're passionate about it, but we also need, a salary, a proper good, security of our life, and so it's harder for those that are maybe, from a lower social economical status to really work for passion. Like this is like, it seems like research, it's the only way to do research is for passion. So I think this is a huge, it's a huge problem for people that actually need the money to to pay rent, and many other things.
Agreed completely. To this I would add, if I look at my own lab, that is the most diverse environment I have seen anywhere, in that I think we have some straight white cis men, but they are by far a minority, and I haven't tried to make it that way. I believe this is because the people who have approached me are people who feel safe in the environment they expect. I'm not saying this to sort of lift the status of the group in any way, but what I mean is that perhaps tells us that if you do feel that you don't fit in among the people that you see, you are less likely to reach out and try to enter a certain environment. This is perhaps not possible to immediately circumvent, because the demographics in one generation then are slower to change. But what we can perhaps do while waiting for the wheels to turn and one generation to turn to the next is mentorship, exactly as was mentioned also, because I can see that in almost every stage where I have succeeded in something, it has been because of supportive mentors inviting me to opportunities, inviting me to collaborate, lifting me up. So what I would like for everyone to do who's involved in the scientific process is to show solidarity, where whoever is not of the mainstream, that is one of us, that is a person we should preferentially support whenever we have a reason, whenever we don't have a reason not to.
You want to add something?
Yes, so if we're talking about research, also at Charité, you showed the numbers. We start with medical students, it's more females, and this is also my experience. I teach at Charité, and then we lose out, right? And especially it's even more in the clinical, on the clinical side, then the gender gap is getting higher and higher, the higher the position is. And this is actually Also, I mean, also in research, like you said, it's not just fun. It's not just for passion. We need the money, and it should be really equally valued. Women and men, and I still see, especially in Germany, there's a lot to do. So with my colleagues, like in the US, for instance, it's different. And especially in cardiology, I just talked to Jutta here before, and it's in cardiology, it's even more difficult. It's very male-dominated. So if this adds to the discussion, I'm happy to add this.
Just want to touch upon one last topic, because I'm wondering, is hiring a diverse group of people, is that just our moral obligation? Because sometimes there's this argument about the tension between scientific excellence on one hand and inclusion on the other hand. And I often hear this argument, basically, when I have two people in front of me with exactly the same qualifications, I don't want to hire basically on diversity standards, basically. Do you have any comments on people that use these arguments in discussion when saying, I just want to hire the best person?
Well, I have a lot to say about that. We just did a conference about this, and it was lovely. Like it's so. First of all, like what is considered excellent is really biased. Yeah. So, for example, it wasn't considered excellent to study menopause or study endometriosis and all these topics that are really affecting millions of women's life. Yeah. So the setting of topics is really not so excellent when it comes to impact. Yeah. So we are not taking the so traditionally so things that affect a lot of women as seriously as we should. But on the other hand, so I do acknowledge that what Sofia has just mentioned and what you have all mentioned, so to manage a diverse team is initially, I think, a little bit more challenging. Yeah, so because they are not on the same page on everything. They may come from different value systems, and for example, also, so we see that at the moment. Yeah, so for example, if you have in your team people who are on different sides of conflicts, yeah, so that can make for a very heated discussion. So and sometimes there is so people who are very similar. They without speaking much, they seem to have an easier way to understand each other. So when you have a much more diverse team, you need to speak more to create this cohesive feeling of we understand each other and I get you. But on the other hand, so I think there's a lot of rewards. Like so, diverse teams can be much more innovative. But as you have said, yeah, so this takes time and takes the resources to really bring people together. So this is not an easy task and for the faint of heart. But I think it's really worth it. And so in a way, I think that diversity is part of excellence. I cannot imagine really a fully excellent team that wouldn't be diverse, I must say.
Just to add to that, I mean, people from different backgrounds will ask different questions. I mean, if you just look historically, when only men were allowed to be doctor, there's a lot of things that could have been solved if they just like asked one woman about something. So we're not going to be able to ask the right questions if we don't include everyone, because we will not be able to think about it. And to what you said, I think what we measure as excellence is super biased. So for a lot of the graduate program, we measure things like the internships people have done, which universities they've gone to, which we know are completely dependent on your socioeconomic background, and it's nothing to do with how good you actually are. And we could do a lot to kind of just remove this to make it easier to hire a more diverse team also. And one difficulty in Germany is also the visa system. It is actually sometimes difficult for us to hire people because they will not get visas. And there's a lot of financial constraints on top of this. Like you have to pay quite a lot of money to have your degree verified. If you're from a country that is not part of a special list, we're not actually allowed to reimburse this in any way. So I think from like kind of the government side, they could do a lot to just help us have more diverse teams as well.
I mean, excellence is a weird and elitist concept, but if we take its premises as starting point for discussion and think of like meritocracy, where we select the best people, the sort of very small, tiny core, etc. and we see an organization that's missing out on some demographics entirely, unless we believe that those demographics are somehow worse at this, then we must conclude that we have failed to find some of those geniuses because they were artificially cut out. So the more excellence you actually strive for, the worse the loss you will have from cutting things out in a leaky pipeline.
I think we could continue for hours on this topic. I feel that there's much more to discuss, but we're also limited in time. And I think we very much highlighted the importance of diversity in science, and also really the need to move towards gender personalized medicine. And I would like to thank all the speakers for sharing their thoughts tonight, the audience for joining. And I would like to remind you that there's a small get-together outside. There's some drinks, so please just stay. All the speakers will be here. If you have more questions, just don't be shy and come ask them. We are very happy to answer. And have a good night.